Hello students, in the previous class we have seen the introduction of the nucleic acid, that is DNA, RNA. So let us discuss about structure of nucleic acids. So structure of nucleic acids. So structure of nucleic acid. Structure of nucleic acid is nothing but it consists of nucleosides and nucleotides. Okay. So coming to here, what is a nucleoside and what is a nucleotide? So here when you take here, nucleoside is nothing but here a unit formed by the attachment of a base to first position of sugar is known as nucleoside. So what is nucleoside here? So when you add a base to the first position of sugar, means it is forming some type of compound. So means what here it is forming. So means what here, base plus sugar, it will give nucleoside. Okay. So means what happening, what is the structure of this? If you take this one here, like this Furano structure, if you have been taken, here it is a first position. Okay, so this is the base, hydrogen, CH2, OH, HOH, HOH. So this is nothing but the nucleoside structure here. So when you take this one, so when you take it this one, what happens here? This is the base. So the base is adding to the sugar at first position. Okay, so that one we call it as nucleoside here. Okay, so coming to here the nucleoside. So here the bond present between sugar and base. So what is the bond present? So this is the bond is present between hydrogen and the base. That one we call it as N-glycoside bond. So what we can say here, the bond present between sugar and base. So this is the sugar molecule. Okay, so the base is attacking. So means what, this one we call it as N-glycoside bond. Okay, so here the N-glycoside bond is between purine and sugar is 9 and 1 position. Okay, so what is the base? So this base, whatever you are adding, so the bond you can between. So if it is present in purine, so if this one is present, this N-glycoside is present in purine, that one we forming as the 9 and 1 position. So if it is present in pyrimidine structures, which we saw in the previous class. Okay, so in pyrimidine means it will form it as 1 position. Okay, so these are called adenosines, guanosine, adenosine, cytidine, thiamine, uridine. Okay, so here ine at the suffix, what you are getting here, ine, nucleoside. So what it is, nucleoside, no? So here what you will be getting, so when you, so there will be some names like that. Example, if you have been taken adenine plus ribose sugar, so this one we call it as ribose sugar. It will give adenosine, uracil plus ribose sugar, it will give uridine, and cytosine plus ribose sugar, cytidine, guanine plus ribose sugar, it will give guanosine, thiamine plus ribose sugar, it will give thymidine. Okay, so this is nothing but what here, this is nothing but nucleoside. So now let us see about nucleotide. So what is nucleotide? Nucleotide is a phosphate ester of nucleoside. Means what here? Nucleoside join. This is a nucleoside which is joined to a phosphate group. That one we call it as what here? Nucleotide. So means what we can say here? A nucleoside joined to a phosphate group is called nucleotide. Okay, so it contains a base and also sugar and 1 to 3 phosphate groups. So means what we can say here? Base plus sugar plus phosphate. We call it as what here? We call it as nucleotide. So the structure will be like this here. Coming to here, the structure will be so here base, hydrogen, HOH, HOH, H, CH2, phosphate group now, O, P double bond O, O minus. So this one we call it as nucleotide structure. Okay. So coming to here, the phosphate group in nucleotide attached to either 5' or the 3' also. So you are having like this 1, 2, 3, 4, 5. So whatever the nucleotide, so it may attack to what here? Either 5th position or also 3rd position also it can attack. So nucleotide can contain connected by 1 or 2, 3 phosphate groups at 5 OH of 1 nucleotide. So means what here? It can be giving as like this. So what I have told you, nucleotide. So this nucleotide connected by 1 or 2 or 3 phosphate. It may have 1 phosphate group, 2 phosphate group or 3 phosphate group. Okay. At 5th position, at 5th carbon, it is going to have 3 phosphate groups at 5 OH of 1 nucleotide. Okay. So means what here? If you have been taken adenine, ribose plus phosphate. So here we are taking purine and ribose and phosphate. It will give AMP. So means what? AMP is nothing but adenosine monophosphate. Okay. And also if you take adenine, ribose plus 3-phosphate is equals to ATP. So ATP indicates what here? ATP indicates adenosine triphosphate. Okay, so here means what we can say? Nucleotides are joined together by phosphodiester linkage between 3OH group and 5OH group of next nucleotide we can say. So dinucleotide contain 2 nucleotide subunits. Oligo means 3 to 10. Poly means more than 10. We call it as polynucleotides. Same like polymers. So means what we can say here? Dinucleotide contains 2 subunits. Oligonucleotides 3 to 10. Polynucleotide means more than 10 we call it as. So here DNA and RNA. DNA and RNA is the polynucleotides we can say here. Nucleic acids. So generally it is composed of long strands, we can say, which consist of nucleotide subunits linked by phosphodiester bonds. Okay, so means what, it is forming different types. So means what, we can say phosphodiester linkage. For phosphodiester linkage is nothing but what, here if you have been taken phosphate linkage, for example, here base OH CH2 O P double bond O minus. So this is one thing. Okay, so this is the sugar I am taking here, molecule, same nucleotide molecule. I am combining two molecules here. Base OH CH2 OP double bond O. Okay, so this is also a sugar molecule. So this one will give, when you combine these two molecules, it is going to give here base we are having, here O P double bond O minus. This one is a 3' end. And coming to here, what you are having, CH2 O P double bond O dash. Okay, so minus. So this is the fifth one. This is 5', this is 3'. So coming to here again there will be attack, CH2 OH H, this is the base. So again 3'. So means what? So this one we call it as, so this linkage we call it as phosphodiester linkage we are going to call. Okay, so means what, here it is going to form like this. So means what it is going to form, how it is forming, a nucleic acid chain is going to form like this, that is sugar base phosphate. So this is a polyno, again sugar phosphate base. So this is a polymer, that's why we are writing like this, sugar base. So this is nothing but a nucleic acid chain consists of. Okay, so here a nucleic acid contains, so whatever you are taking, so the nucleic acid chain is abbreviated by one letter code with 5' n, and it is written like this. So phosphate here, this is 5'. Coming to here, A adenosine 3' phosphate, C cytosine again 3', 5'. So it is going to link like this, phosphate again 3', here guanine 5', phosphate 3', uracil phosphate 5', thiamine. So like this it is going to end. Okay, so like this, so means finally it is going to get OH. So this is the way it is arranging. Okay, so now let us see the structure of DNA and RNA. Structure of DNA and RNA. So generally DNA, so DNA is nothing but what here, DNA is nothing but here, let us see, first of all DNA is nothing but deoxy ribose nucleic acid. Okay, so here it is a double helical structure which was proposed by Watson and Crick on X-ray diffraction studies. What we can say here? It is a double helix structure. Double helix structure proposed by Watson and Crick based on X-ray studies. Okay, so means what here? It is like a spiral cord ring we are going to have. Okay, if you have been observed here, it is like a spiral cord. So means what here? It explains basic base equivalence and duplication of DNA. Means all species. So it contains A to Z and C to Z. So here total amount of purine is equal to total amount of pyrimidine, we can say. These two are purines, these two are pyrimidines. Yes or no? So by this what we can say? Total amount of purine is equals to total amount of pyrimidine. So in the previous class, if you have been observed, so there will be structures of purine and pyrimidine. So the purine is nothing but A plus G is equals to C plus T. So coming to here, the double helical composed of two right-handed helical polynuclear strands coiled at central axis, same central axis. Okay, so the two strands which are parallel with each other, and there will be having 5-3' phosphodiester linkage with opposite direction. So what we can say here, the double helix composed of two right hand helical polynucleotide strands coiled around the same central axis. Okay, so means I'll show you the structure, and also so here one more thing we can say here, the two strands what you are taking, the two strands are anti-parallel. The two strands are anti-parallel with each other, and their 5-3' phosphodiester linkages opposite direction. So means what here, if you have been taken the structure of DNA, so those are anti-parallel, and also the positions when you link the phosphodiester. So if you have been seen phosphodiester linkage, so it should be in the opposite direction. So here the base groups whatever present, that will be in the inside helix and perpendicular to helical axis. So the base is nothing but here adenine, guanine, cytosine, these present in the inside. The base groups are present inside the helix and perpendicular to the helical axis, and also the two strands. So the two strands what we are taking, they are held by hydrogen bonds. So means what, the two strands which are bonded to hydrogen bonds, held together by hydrogen bonds, that is we can say here A is equals to T and G is equals to C. Okay, so A forms with hydrogen bond with T, G forms 3 hydrogen. So this will form 2 hydrogen bonds with T, this will form 3 hydrogen bonds with C. Okay, so here A does not form. So A does not form with C and G does not form with T. So the length of all these hydrogen bonds what you are taking, it is similar, and DNA rotates at both sides, that is right hand side and also left hand side. So here the right hand helix be more stable, that's why we call it as D conformation. Okay, right hand side is nothing but D conformation we are going to see. So it will rotate at what we can say DNA rotates at both sides, that is right hand side or left hand side. Okay, so the right hand helix is more stable, so that's why we call it as B conformation. So now let us see about the diagram here. So it is going to have like this. So double helical structure now. So I'm drawing. Okay, so here you are having 5' phosphatase linkage, 3', and coming to here, this is the 5', this is the 3'. So inside your, what I have told you, these two are the strands which are anti-parallel. So the phosphate linkage, whatever it is present, it is at opposite ends. Okay, and coming to here inside, this inside we are having A is equals to T, G is equals to C, A is equals to T. Like the base pairs, whatever base pairs we are taking, so that is in the inside we are having C is equals to G, C is equals to G, A is equals to T, A is equals to T, C is equals to G, A is equals to T, and also here also A is equals to T, C is equals to G. So like this the base pairs are having. So these are polyamines. So amino acids are linked like this, lysine, glycine. Inside the strands we are having lysine, glycine. Okay, so these are having bond pairs. So these amino acids, whatever amino acids we are taking, so these amino acids are present inside the strands. So inside the structure, inside the helix, we are having the base pairs. So here it will rotate both sides. It can rotate at left side and also right side. So the right side, so whatever you are taking here, so this is the right side we have considered mostly. Why? Because it is having more conformation. So these base pairs will form hydrogen bonds with amino acids, lysine, glycine, leucine, isovalent. So these amino acids are forming hydrogen bonds. So A can form 2 hydrogen bonds with T and G can form 3 hydrogen bonds with C, but these cannot form with A is equals to G, T equals to G. Okay, so this is about the double helical structure. And in the secondary structure, if you have been taken RNA, so coming to here RNA, so here helicals are present only single stranded. So RNA is nothing but what here? RNA is nothing but here ribose nucleic acid. So ribose nucleic acid, we call it as RNA. So here the helix are present what here? The helix are present only single stranded. Okay, sometimes they fold back on themselves and like a heparin. So like our backbone, they acquire double helical structure based on the double stranded. Okay, so in double stranded arrangement, what happens? Guanine pairs with cytosine, adenine pairs with uracil. Okay, so coming to RNA, so if you have been taken, the structure will be like this. You are having CG, CG, GC. RNA contains uracil in place of thiamine. Okay, so it is going to form like this: UZ, CC, GC, UA, UA, UC, UA, UZ, CG, GC. So these are having like this 5' phosphate, 3'. So it is forming loop, we call it as here. So these all also having bonds. This one we call it as stem. How the tree is having the stem to form the leaves, in the same way like that it is going to have the linkage ribose. So here what we can see in double helical structure, RNA molecule. If you have been taken RNA molecule, it is a single stranded, we can say. Why? Because only one of the two strands of a gene we are having. Okay. So coming to here, guanine contact does not, it is not, so guanine is not equals to the cytosine contents, and also adenine contents will be equals to uracil. A equals to E, cytosine is equals to G. So means RNA is hydrolyzed. There is no relation among the quantities of different bases. So they itself only will undergo hydrolyzation. So there are three types of different functions. They are named messenger RNA, we can say here. Messenger RNA, ribose, that is mRNA, and also here we are having ribosomal RNA, that is rRNA, and transfer RNA. Transfer RNA, that is tRNA. Okay, so this is about RNA, we can say here. And we are having fingerprint. So fingerprint of RNA we are going to have. So what is fingerprint? So fingerprint of DNA fingerprints, we can say. So DNA fingerprint. So each human has unique fingerprint. Okay. So there are the tip of the fingers which will identify that human. So they may be altered due to surgery also. Okay. So what we can say, each, if you have been twins, we can't identify. Based upon their fingerprints, we can easily identify. Why? Because each human has unique, their own fingerprints they are going to have. So they are at what here? They are at the tips of finger, useful to identify that human. Okay. So if we can't identify that human, we can useful to identify. To avoid this DNA fingerprinting, they have introduced, means what here has been introduced to identify the human beings also. So DNA fingerprints is based on the sequence of bases on DNA. So these are based, so what we can say, this is based on bases of adenine guanosine, because of this DNA is unique for a person. So okay, this is based on adenine guanine, because of that only we can identify a person. And the sequence of base is the same in each and every cell and cannot be altered by any known means. So means what here, this sequence of base is same in each person. Okay, it will not be same in the different persons. So what we can say here, sequence is the sequence of bases is the same in each and every cell, we can say, and cannot be altered by known means. So means what we can say, the basis whatever sequence we are taking, it is same in adenine, guanine, cytosine, uracil, whatever you have been taken, the sequence will be the same, but it cannot be different by with other person. So the DNA fingerprinting, where it is useful, it is useful for identify the criminals by forensic labs. Okay, useful for what here, identify the criminals, by forensic lab, and also determining the paternity, so paternity of the individual. Sometimes they may cheat their parents. Okay, and also they can determine the paternity of individual, whether the children is the parents or not. Paternity of individual, and also dead bodies by combining the DNA with parents or children's. Okay, so dead bodies can be can be compared by parents or children. So, and also here to identify racial groups to rewrite biological evolution. For example, sometimes they may sign their signatures. For that also it may be biological evolution. So for this also the DNA fingerprint can be used. So this is nothing but what here? So this is nothing but DNA fingerprinting. So I have told you here messenger RNA, ribosomal RNA, transfer RNA. So coming to here replications, we are going to have transcription, we are having annealing. Annealing is nothing but here when the melted DNA is cooled, the strands hybridize. That one we call it as annealing. So here if you have been observed here, what we can say here, when the melted DNA is cooled, the strands hybridize. That one we call it as annealing. Okay, so this is annealing. Annealing means so the DNA what you are taking, so it is cool, it is in hot condition. When it is cool, the strands whatever you taking, it will hydrolyze. Okay, hybridize. That one we call it as annealing here. And also replications means nothing but means here the synthesis of identical copies. The synthesis of identical copies of DNA, we call it as replication. So means what here? Nucleic acid control hereditary at molecular level, and the two strands of DNA what you are taking, they are complementary also, both them. So they will carry same genetic information. Okay, so the genetic information is controlled, it is having same genetic information. That one we call it as replication. So that one we call it as what here? New daughter DNA molecule identical to the original. So means what? That one we call that they will carry original genetic information. Coming to transcription. Transcription is nothing but here the synthesis of mRNA from a DNA blueprint. The synthesis of mRNA from a DNA blueprint is called transcription. So this will be taken under the nucleus of the cell. So it provides fingerprint, we can say synthesis of DNA of blueprint, we can say, which we call it as replication also. RNA is much shorter than DNA. Why? Because here we are having messenger RNA,